They are the invisible killers. No matter how much we try to evict them from our surroundings, these deadly microscopic organisms that are lurking around us have the ability to wipe out an entire population in an instant. Influenza alone causes 500,000 deaths and approximately 1 billion infections a year. Nearly 500,000 to 1 million people die of malaria every year. These germs, viruses, and parasites form diseases such as HIV, Ebola, influenza, and malaria. One such invisible killer is the malaria parasite. Malaria, transmitted from mosquitoes' saliva into the host's bloodstream, is one of the most lethal and infectious diseases on the planet.
Nearly 500,000 to 1 million people die of malaria every year.
Professor Peter Preiser has been working with the malaria parasite since 2003.
So the parasite causes the disease by living inside a red blood cell, so it invades a red blood cell, it grows inside a red blood cell, and then after 48 hours, the red cell gets ruptured, releases new parasites, and they continue that cycle. And that goes on and on until either the person survives or dies. In the sense, it is a cell which is just the same as our human body cells are made out of, which makes it often very difficult to target by drugs. So antibiotics don't work. So ultimately, it means how ill a person gets. That's a big starting point for trying to think about intervention.
Clinical intervention can be done in the form of faster diagnostics. Professor Peter Preiser and his team have developed a new diagnostic tool for faster detection of the malaria parasite.
We've been able to work and develop a new diagnostic tool. And this is based on the fundamental discovery that when the parasite lives inside a red blood cell, it eats the hemoglobin in the red blood cell. And when it eats the hemoglobin, there is a metabolic byproduct which contains iron. And this iron, when it's normally in a red cell, has no magnetic properties. But after it sort of gets metabolized by the parasite, it becomes what is called paramagnetic. And that is something that you can detect in a magnetic field. We can take advantage of this minute shift in the magnetic properties of the infected red cell to detect it at a very, very highly sensitive approach. So here we've shown that we can detect amounts of parasites which are lower than any of the standard techniques that we're using, and that it's also easier and faster to use.
Not far away is a deadly virus that affects us through our respiratory system. They are airborne. They are called influenza.
Influenza is very unusual in that it has what we refer to as a zoonotic cycle. So a zoonotic cycle means that the virus can replicate, infect, and transmit amongst animals. And in this case, what happens is some of these viruses actually transmit to humans.
In 2009, the world was struck with the H1N1 virus, infecting more than 1 million people.
H1N1 can be from a variety of different sources, and the last one which caused the pandemic was a reassortment between viruses that have chicken, swine, and human origin. And this reshuffling of genetic components gave rise to a unique virus that was not detectable, which was unique to the human population, and that was caused the pandemic. Because it was a new virus that no one really knew how this would progress, what the level of severity was, we were never sure that whether it would be mild or whether it would kill a lot of people. So actually at that time when we were working at this agent, there was a lot of uncertainty. We were able to produce one of the first back-to-back comparisons with laboratory isolates with avian viruses which were isolated in Singapore. Cells have pathways that are activated during infection as a protective measure, and so what we were trying to look at is whether these pathways were activated by the pandemic virus, and indeed they were.
These intangible microorganisms that roam amongst us will continue to spread like wildfire, infecting friends or foes. With these virus hunters, perpetual researchers and discoveries, It enables us to be less susceptible as victims of a pandemic.
The pandemic virus is evolving, and no one is quite sure what the end product will be. There are new viruses which we work with, H7N9.
Because where Singapore is, it's always at risk. The vectors exist, they can easily replicate here. It's a perfect climate for malaria. If you let control snacks, malaria comes back very, very quickly.
Pandemic H1N1 will never be eradicated.
I'm more optimistic. I think it is possible. Malaria isn't a big problem in Singapore.
When everyone was looking at H5N1, pandemic H1N1 came along, and that was a surprise. So it could be that something else will come along that we are not expecting. In fact, it's very likely. H7N9, which is still ongoing, that came along. That was completely out of the blue. No one knew this.
For malaria, it needs to constantly control mosquito populations. So it's only as long as you keep intervention going will you prevent malaria from coming back.